My name is Mark Hallett. We are at the Movement Disorder Congress in Dublin in 2012, and I have the pleasure and honor of interviewing Warren Olanow, who is a past president of the Movement Disorder Society, and we're going to talk about his career in movement disorders and all about his interactions with the Movement Disorder Society and so on. So, Warren, the first thing that would be worthwhile telling us about is your early life, how you got interested in medicine.
How I got interested in medicine?
Right.
Well, I wasn't good enough to go into mathematics and physics, so I turned to medicine. I always loved medicine because it really represented the complement between the humanistic issues I enjoyed and the science, and it was a chance to bring them both together.
So, but where did you get the idea of medicine as opposed to perhaps biology or neuroscience?
Well, when I was young and in school, my main interests were in mathematics and in physics. When I moved into educational higher ground, quite candidly, it just seemed practically mathematics and physics was a difficult road to pursue, not many opportunities, and that medicine would give me an opportunity to exercise science. And it is interesting how mathematics and physics are becoming more important in medicine today. And at the same time, quite candidly, it did allow for issues like humanism, philosophy, literature to blend and allow you to do great scientific things and to help people at the same time. And I think in a much more immature way, that's what I sensed as a young person.
And so where were you then?
I grew up in Canada. I went to school at the University of Toronto and went to medical school at that school.
And so you got interested in medicine.
Yes.
And then somehow you got diverted into neurology.
Well, neurology to me was easy. You know, I went through a period, and I think many people have gone through it, that when you're going through medical school, there were so many areas of interest that I had, and it was difficult to be sure what to choose. But I instinctively knew the nervous system was what I liked. The tough part for me was, should I be a neurosurgeon, should I be a neurologist, should I be a neuroscientist, should I... That was the difficult part. It was not hard to know I wanted to do neurology. It was the most internal of internal medicines.
Right. So how did you pursue that? I mean, what did you do to follow up your interest in that regard?
Well, I ended up doing a residency, and it was interesting because in Canada, most people in my day and age stayed home. They didn't even leave their city to go to another city. All your education was in one place, and then you typically stayed at that institution or that city for your career. I had a teacher who was one of my mentors, who had trained at Columbia University under Houston Merritt. He had been inspirational to me as a student. He was a wonderful neurologist, great teacher.
And who was that?
This was a man named Joe Murata, and it's very interesting because there are many wonderful neurologists that have come out of Toronto, and I think others, such as Tony Lang, would tell you the same thing, that Joe Murata was a role model for him as well. In my case, in Tony's case, he attracted him to stay. In my case, he encouraged me to leave. He strongly recommended that I think about going down to Columbia and training at Columbia, which I ended up doing. So I ended up training in neurology at Columbia in New York, a decision I never regretted. And
so who were the leaders at that time at Columbia?
Well, Houston Merritt was still there. He had become dean of the school. There were many, I mean, Stan Fawn was just recycling back, so I had the opportunity to interact with him. But the people who probably had the biggest impact on me and influenced my subsequent movement into movement disorders were Melvin Yarr and Roger Duvoisin, and many of the wonderful scientists that they worked with.
So what sort of work were they doing at that time that got you interested?
Well, they were busy learning about dopamine and levodopa and how it works. And you know, what's very interesting, Mark, at that time, the idea that dopamine deficiency could be replaced by levodopa led to the idea that there would be other neurotransmitter deficits that accounted for diseases like Alzheimer's disease. And it's interesting that I may be wrong, maybe you can correct me. I can think of only one other disease that really turned out to be helpful to, and that was myoclonus, where a serotonin deficiency was recognized and they replaced it and it helped. But at that period of time, it was thought we could accomplish things. I'll tell you something that was very instrumental to me as well, and I think changed the field. And that is that in neurology in particular, but I would say in medicine in general, almost every drug and almost every discovery was by serendipity. And one of the points I wanted to make, Mark, that was so interesting to me is that the vast majority of drugs and treatments discovered up until that time were discovered by serendipity, not by premeditated science. Perhaps the first, certainly one of the most important in our field, was the rational discovery that there was a dopamine deficiency in Parkinson's brain and that the precursor could be used to try and replace it, and it worked so well. And that, of course, gave hope that all these other neurological diseases potentially could be solved at a laboratory level. You wouldn't have to just wait for serendipity. Turns out serendipity is still a big promoter of good drugs for us, but it was a pretty inspirational concept at that time.
Mm-hmm. So it clearly got you excited to be working with those guys, and at this particular moment. So how did you follow that up? I mean, what was the next step in your own career that sort of related to all that?
Well, the next step for me is after I finished my neurology residency, I worked with a neuroanatomist, Malcolm Carpenter. Malcolm was at Columbia. He was one of the leading neuroanatomists of his day, and I had the opportunity to work with him on the basal ganglia system. It was a wonderfully interesting time. They had already discovered that the nigra and the striatum were linked by dopamine, but the precise anatomy had still not been worked out. The nigrostriatal tract, as you know, is a very small, tiny little tract. It was not visualized with stains that were used in those days, so people had to invent new methodologies to be able to see them. All of that evolved, and one began to get a sense of how the basal ganglia was put together, the role of the subthalamic nucleus, the idea that you could get movement disorders by lesioning the subthalamic nucleus. All of that was taking place. It was a very exciting time. Very meticulous. I don't know if you've, I'm sure neurophysiology is equally meticulous, but when you're in a laboratory setting, clinical is a bigger, more global, more broad, sit back. Sometimes people don't realize that in the laboratory, especially in fields like anatomy, physiology, biochemistry, it's very time consuming, very meticulous, a lot of work and effort.
Yeah.
Worth it, but...
Right. One of my first papers was in neuroanatomy, and I got headaches looking through a microscope, so I went into physiology.
Well, I got shocked in physiology, so I went back to anatomy.
Right. So
one of my other heroes when I was a student there was Sid Gilman, who was doing physiology at that time and making wonderful discoveries. And, you know, quite candidly, I went to look at Sid's lab when I was deciding where to go, and Sid had already taken on his complement, and I ended up working with Malcolm. But it is interesting how our careers might move in different directions, but for quirks of fate.
Right, so carry on with the story. What happened next?
Well, I finished my training. I finished my postgraduate training in neuroanatomy, and my first job was at McGill at the Montreal Neurological Institute, the Jewish General Hospital in Montreal. I began to see patients. I did neurology. I began to do some clinical research, and I taught the neuroanatomy course, neuroscience course at McGill. So that was how I moved forward. I did a couple of studies in those days, but mostly clinical medicine. In retrospect, that was very good because I got the opportunity to see an enormous amount of clinical medicine and began to slowly specialize in movement disorders, which at that time was not a true subspecialty.
Right, I was going to say at that time it wasn't a true subspecialty, but you obviously were interested in Parkinson's disease, so that was, I mean, did that become a focus particularly?
Yeah, Parkinson's did become a focus for me, and I set up and ran a major Parkinson's disease clinic. It was a wonderful experience for me, and it's interesting. We had an aged home there, a retirement community where there were many Parkinson patients that I would look after and do studies on. And I remember it so well because there was a huge Russian community who were elderly and had these diseases, but oftentimes were smart and remembered things. And I remember they had lived through the Russian Revolution. So it was one of those wonderful moments where you could get a sense of history, living history, from these people.
Right, but then you didn't stay there forever. You moved back to the United States.
I did. Montreal was going through a difficult period of time. I must qualify and say Montreal is one of the greatest cities I've ever lived in. I just loved Montreal. I learned to speak French, so to speak. There was a wonderful group of inspirational people there in science. One of my mentors there was Joe Martin. But the politics began to intrude on the operations. And many really good people decided to leave, including Joe Martin. At that time, I had been recruited to go to Duke University, and Joe, who had at that time gone back to Mass General, strongly encouraged me. He felt there just wouldn't be support and funding in Canada at that time. So I ended up going to Duke.
And was it there that you got interested in MRI?
Yes, I worked with Burton Dreyer. We had the good fortune to have one of the first high field strength magnets, and we recognized the iron effect, the low signal on T2 weighted image, and we recognized it was mostly in the basic ganglia. We were able to ask things like, what's it doing there, and figure out that it was iron. And
so what did your interest in neuroanatomy lead to the MRI? Is that, I mean, that's an obvious link, I suppose.
Well, in a way, in a way it was, even more so now, because as you know now, you know, with the quality of MRI available and the very, very high field strength, you can literally do living anatomy now.
So that was the main type of research that you were doing when you were at Duke?
Yeah, in Duke, I was head of clinical neurology, and most of the work I did was clinical. I ran a Parkinson Center, a movement disorder group. We did do some clinical trials, but I had broad experiences. But what happened to me at Duke, and it's an interesting thing, and I don't know, Mark, that it's possible or even wise to contemplate doing it today. But here I am now. I've graduated from McGill. Excuse me, I've been on faculty at McGill. I've now gone to Duke. I've now been in practice quite a few years, and I suddenly realized that my real love is in understanding these diseases better and bringing that information back into the clinic in experimental therapeutics. And to do that, I'm going to have to be a clinician and go do some basic work and work in the laboratory. At about that time, we had discovered iron. It's funny how things look back. We, in our earlier discussions, have talked about once you know something, how easy it seems in retrospect. But for me, I was just learning about iron and Fenton chemistry and that it promotes oxidative reactions, and the idea that dopamine generates peroxide, which reacts with iron to form hydroxyl radical, was pretty radical to me, and raised the idea that this might be the cause of Parkinson's disease, and I had to go and study that. I had to. It was like I couldn't not do it. Right. So I was unlucky in that I never really had the kind of formal science background I would have liked and had a career in science to parallel the clinic, but I was lucky that I was looking at something at such an early time that there was a lot of low-hanging fruit.
So how did you pluck off the low-hanging fruit? What did you do?
Well, we began to do studies. We began to see that iron was increased in the nigra in particular of Parkinson's disease patients. We began to see iron was increased in the putamen of patients with MSA. And we began to speculate that iron might be the primary cause. We did experiments. I injected iron, and the iron would created a syndrome. It would replicate the Parkinson features. We were pretty excited until one day we did an experiment where we cut the median forebrain bundle remote from the nigra and then watched iron accumulate in the nigra and recognized for the first time that iron accumulation is a secondary phenomenon that occurs in any form of neurodegeneration, and that all the iron we're seeing could have been nothing more than just a marker of where degeneration was occurring. I mean, it was funny. It was disappointing on the one hand. It was exciting on the other.
But again, you didn't stay at Duke.
No, because I wanted now to be more involved in basic research, and Duke was such a wonderful institution. And they had so many great scientists there that the idea that I, who was head of clinical neurology, now wanted to also do laboratory work was not necessarily looked on as being a serious endeavor. At about that time, I was recruited to the University of South Florida, where I was offered the opportunity to set up a Parkinson and movement disorder clinic, and was given substantial laboratory space, interestingly, right next to John Hardy, speaking of the world being small. Actually, I was there before John, and John was recruited when I was already there.
So what was the state of movement disorders at that time? The field was already beginning to develop a little bit more clearly. When you say Parkinson's disease and movement disorders, it sounds like it's already beginning to develop as a field.
You know, Stan and David had already begun to bring things together. It was around that time that the Movement Disorder Journal was being established, the societies were being established, the phenomenologic aspects, I think, were being brought forward mostly by Stan, and the translation into the scientific basis really by David. I mean, they were a wonderful combination, and people really began to try and understand these movement disorders better in the clinic and in the laboratory. I mean, you may not recall, but just to show you where we were at that time, I remember one of our major meetings. I believe it was run by a gentleman named Mark Hallett, in which people were trying to define what myoclonus is, and you actually took a vote of the audience and you ran a series of slides in which you timed things and said, okay, how about this, 200 milliseconds, 150 milliseconds, 100 milliseconds, and at what point do you call it myoclonus? And I remember the vote was 100 milliseconds, and for all I know, that's how the definition got made. Do you recall that?
Yes, that was the Washington meeting in 1990, where you and I worked very much together developing that particular meeting, and that particular session came from a concept that Paul Del Wade had proposed on trying to deal with what normal phenomenology was of different things. And the question was, what was a quick movement? And so that's how we got into the issue of the timing of myoclonus. How quick is quick was the issue for that particular one.
Every era has its excitements for young people going into the field and what they can look forward to. But looking back, how wonderful it was to be there at a time when we defined the UPDRS. I mean, everybody takes it for granted. I mean, we struggled hard. You know, it happened to be at a meeting in Bermuda over cocktails, but nonetheless, we worked hard to develop these scales. And to think that you're defining myoclonus, that these fundamentals are being, how do you define dystonia? We're still arguing what is Parkinson's.
We're still doing it. We're still doing it.
But it is fascinating to think that we were back there. Historically, it was a very interesting time, though, because as you may recall, the movement disorder world, which was still in formulation at that point, had really divided itself into two groups. One was largely a European-based group, which was called the ISMD, the International Society for Motor Disturbances, I think it was called.
Right.
And they held a meeting, and that meeting was very successful and growing rapidly, in which information was transferred and communicated. And that was largely a European society. And on the other side of the ocean, there was the Movement Disorders Society, which was founded and organized and largely was centered around the journal, which had just been introduced. And at that time, the journal, if I recall, was coming out perhaps every 3 or 4 months, something like that, maybe every 6 months at the beginning.
Well, it was probably I don't remember exactly. It was probably every 2 or 3 months, I think.
Well, no, I think there were at most 4 issues a year at the beginning.
Yeah, right, 3 months.
And then at the other hand, we had our meeting, and if I recall, the international meeting was every 2 years.
Every
roughly.
2 years, right.
So that was the beginning of the beginning. And this society started to grow, and this society started to grow, and an awful lot of the American members joined the ISMD, and a lot of the ISMD members joined the Movement Disorder Society, so that it became inevitable that at some point these societies should merge.
Right, right. So anyway, let's go back to Florida for a bit.
Back to Florida,
Back to Florida, and tell us a little bit more, I think, just sort of about the main research type things that you were doing there, because you've been very active in a variety of research things. It would be nice to sort of chronicle your own career in that regard.
Well, fundamentally, I'm a clinician, Mark, and everything I've done is clinically oriented. And so I may have gone into the lab to look at why cells die, or to look at how we can fix them or restore them, but it was always with an eye to the clinic. So I was very clinic-centric in the way I approached it. We did work looking at oxidative stress. In those days, we did work trying to understand the role of iron, trying to understand the role of glutathione. I remember we put together and we published as a theme in Annals of Neurology a giant meeting where we brought together everyone who was interested in these areas. It was, in my mind, a very seminal meeting where the idea was to bring together people who were in the field working in these areas like mitochondria, like oxidative stress and so on, and bring them together with people who were not in the field, but who were world leaders in outside areas. That, by the way, remained a tremendous theme for me. And just to jump forward in my current life, where I'm very interested in proteins and prions, what I did then and what I implemented now was I learned that by having that kind of meeting and bringing together people who were world leaders, but in different fields, but not in movement disorders, but in your area, you could fast track your own work tremendously. So I remember talking about iron, and in those days we knew about iron 2 and iron 3, and we were, you know, the idea was that ferrous iron stimulated these reactions. Ferric was the more stable form that was stored as ferritin, and this is what we worked on. And we brought in an industrial chemist who specialized in iron, and he said, you know, none of your experiments really mean anything. And we looked at him and said, why not? He said, well, have you considered iron 4, 5, and 6? And we said, there's an iron 4, 5, and 6? He said, yes, and they're the ones that are really, really reactive. So if you get contamination with iron 4, 5, and 6, it could explode your reaction. It's much more promotive of these kinds of reactions. And by the way, you should be doing these like in a spacesuit, in you know, infectious garb, so that you can't contaminate. That's the kind of thing you were able to learn right then, rather than doing 5 years of research and finding out it wasn't reliable. So those were the themes of my work. That was also the time when I began to become interested in transplant. I worked with Tom Freeman there, who was very interested in it, with Jeff Cordover, and together we began to put together the nidus of doing a transplant program that we would try to see if we could replace dopamine neurons by implanting embryonic dopamine cells into the striatum. And it was great fun because we did so much of the preliminary studies to determine at what age the embryonic donors should be and how many we should use and in what form we should use them. So that was several years of work to get to the point where we were ready to move forward to clinical trial.
Right, and that was, of course, a very important trial and a very important part of the development of Parkinson therapeutics.
Right.
Yeah. Right, so carry on. I guess it was around that point that you again moved from Florida back to New York City.
Right. It was around that time, and it was very interesting because I had just been recruited at that point to be chairman of neurology at Mount Sinai in New York. And while I loved it in South Florida and had put together wonderful collaborations in pharmacology and with Tom and others, the opportunity to come to Mount Sinai to replace Melvin Yar, who was still live at that point, still working, still my colleague and friend, that was very exciting, and I could not resist that opportunity. It was very interesting, though, because the importance of granting and grant funding was just, we were going through that change when, in the early days, I don't know if you see it the same as I do, but there was a time when clinical medicine seemed to drive programs and research lived off clinical revenues. And that transition was just happening where clinical revenues were now coming down. It was hard, especially for academic clinicians, to be able to be self-sustained. And the academic research grants were what was driving a clinical department. And we had put in a very large NIH grant to fund our double-blind transplant study. And literally the week I went to Mount Sinai, I got what probably was the largest grant that Mount Sinai had had. So at least for that 1 week, we were really in good shape.
Right, right. So then you've been at Mount Sinai since.
Yes, I
have. And maybe just a little bit about what you consider to be the most important things that you've done during that period of time. You've been involved in lots of different things.
I have.
Let
me preface it by saying that when you become a chairman, I really believe that the critical decision you need to accept and be willing to take on is that other people's work become as or more important than your own. And if you ask me what I'm most proud of, it would fall into that area. So I didn't stop doing my own work, but I'm extremely proud of the brilliant people and the tremendous number of young people that we trained, that we work with, that have gone on, many of whom have gone into movement disorders. And it's a joy for me to come to meetings and see so many of the people that I worked with who are now leaders in the field. As for myself, we carried out the transplant study, and we had great fortune. We were the first group to show that implanted cells could survive and reinnervate the striatum, which gave credence to the idea that transplant could work. We did a double-blind controlled trial, and as you know, sadly that study failed. But I think in looking at it in retrospect, and as we reported in our articles, if you look at the subset who were younger, milder, had more pure dopaminergic lesions, they did do well. So, and there are studies now underway where people are looking at transplants still, and of course for stem cells this will turn out to be very important. And I think there are a subset of patients where the concept of transplant will work if in fact that's still necessary. So we did that. We were one of the first groups to recognize diphasic dyskinesia. I know Paul Green at Columbia and his colleagues reported it, and then we found that it was present in as many as half of our patients. So that set out the importance of diphasic dyskinesia as a limiting factor, and we obviously have done work, and others have, to try and understand why they occur and how to avoid them, which will be critical to moving the field forward. We made the extraordinary discovery that in a patient who died after transplant, that Lewy pathology had now developed in the implanted neurons, and that fostered much of my later work, which has been based on protein accumulation and why proteins accumulate. We discovered that the proteasome system was defective. We thought that there was an accumulation of protein. Alpha-synuclein stories showed that if you accumulate by gene replication, that too can cause the disease. So we were very high on the idea that proteins were at the heart of Parkinson's disease. And now with the idea that they could move from cell to cell, potentially, to account for this observation. The whole idea that Parkinson's was a prion disease came to my mind. I remember the day that paper was published, I got a call from Stan Prusiner, who said, "That's it, Warren, it's a prion disease. There's no other rational explanation for this." And we've been, of course, very actively exploring that, and that remains a major focus. But one of the things I want to make a point, Mark, if I can, for this purpose, that I think is really important for clinicians out there. Two of the most important things I've done in my whole life that I believe have really moved the field have been on single individual cases well studied. One, a patient who had a transplant and a post-mortem showed that the cells could survive and reinnervate the brain. Can you imagine the beauty of seeing that for the first time? Seeing healthy-looking cells sending fibers out, forming organotypic architecture in the brain. How great that was. One case, that's all it took. I mean, there have been others since, but all you needed was one to show that. and then one case showing the proteins could move, or at least accumulate in implanted neurons. All you needed to see was one to know. And I say that because in a day and age where we focus so much on the lab, so much on genetic studies where you need 100,000 people to get meaningful results, a brilliant clinician with careful watching of individual patients can still make contributions.
Now that second observation was perhaps in the realm of serendipity again, right? Because you weren't anticipating that, were you?
Was I anticipating that Parkinson's would be a prion disease? No one believes it even still.
Right, but that is, I mean, it goes back to your earlier theme
Yes.
about serendipity, about being important.
You know, it reminds me of Arvid Carlson, who won the Nobel Prize for his work that led to the development of levodopa. And I remember him saying at a meeting one time, almost with a sad face, that it was no big deal. I just stumbled on it. I just stumbled on it. And I remember saying to him, it may not be a big deal, and you may have just stumbled on it, but it takes a pretty smart guy to figure out what he stumbled on.
Yeah, well, that sort of chance favors the prepared
Exactly.
mind in that regard.
Right.
Yeah, sure,
Right.
sure. So tell us a little bit more about the Movement Disorder Society, your role in the Movement Disorder Society, and what you think the Movement Disorder Society has given to the field.
Well, maybe I should start with the latter first, because that's closer to my heart. You and I have both been presidents of this wondrous society, and at least at this early stage in our history, I've been struck by the fact that it seems to me that the right man has been president at the right time, each of us bringing different skills or gifts to the evolution of the society until it's in its present state, which is just marvelous. I like to think that patients all over the world have received better care because of what we've done. I like to think that we're closer to finding better treatments and cures than we would be without this society. I think we are clearly the major forum for education, for transfer of ideas, for collegial interactions, for people uniting in their thoughts and stimulating, catalyzing new ideas. And I like to think that this has been one of the most glorious successes of our lifetime. As for me, again, maybe it's serendipity, but somehow I ended up as an American, Canadian, but American, I ended up president of the ISMD, the European Society, and David Marsden, a European, ended up president of the Movement Disorder Society. And as so many fine things in life, while we were having a drink together, we suddenly realized there would never be a more opportune time to do this, when all these differences would be put apart. I, on the European side, as an American, wanted this, and he, as a European on the American side, wanted this. And that, I think, led to a meeting that I think you ran, or at least participated in which you began to put together the rules and how this would come about.
Right, right. Was that the time that you guys swapped your credit cards by accident?
You know, it really is a wonderful story that should be recorded. We were out at one of our famous and notorious dinners. We each put in our American Express cards. When the dinner came back, we took them back, not noticing that I had taken his and he had taken mine. We proceeded to use the credit cards for about 3 months without noticing that we were each using each other's credit card. The reason I noticed is I had rented a car and I had taken it back, and the young woman who was at the desk when I took it back, as I was leaving, said, "Thank you very much, Professor." And I looked at her and I said, "How did you know I was a professor?" And she said, "It says so right here on your card." And I looked at it, it said Professor David Marsden. So I called David, and David had that wonderful laugh, that uproarious laughter. And when he was laughing, he heard this, and I said, "We have these cards, so we'll send them back to each other." So we said, "Well, how do we deal with this?" And I said to him, "You know, David, I don't want to go back and calculate all the charges, and it's a pain. Why don't we just say that, well, whatever they are, they are. And if you spent more than me, fine. And if I spent more than you, fine, and we'll just let it be. And that was our agreement. And about a week later, I got a letter from David. Dear Warren, I just want to thank you for the many, many wonderful meals that you treated me to in the fine hotels and the beautiful establishments I stayed at. It was so good. And I wrote him back saying, David, it was a pleasure. And by the way, the car is driving beautifully. Right. So that was a wonderful era of history, yeah.
Yeah. So I guess a lot of your work has been in clinical trials.
Experimental therapeutics.
You have been a real leader in that area. Do you want to say a word or two about that? And I mean, in terms of running clinical trials, I mean, worldwide, you're really one of the experts, the leaders, the organizers of all that.
Well, first of all, in the final analysis, I'm a clinician. In the final analysis, if we're really going to move the field more than incrementally, it will take experimental trials. So I still have an interest in the laboratory, still work there, but my main interest now is in moving the laboratory into the clinic. Obviously, we did the double-blind transplant study, which was extremely complicated. More recently, we've done gene therapy studies, which are extremely complicated. More recently, we've just completed a double-blind, double-dummy study of continuous intraduodenal levodopa infusion. And we've studied many putative neuroprotective agents. We did the first major delayed start design study as a primary endpoint trial. So yes, I think you have to do it, but it takes some courage because these are new, they're novel, and success is by no means guaranteed. In fact, I am frequently known as the master of the failed trial. And you have to be willing to live with that level of failure. But that's the only way I think we're going to move forward. We've not only tested a lot of new and exciting therapies and continue to. We're currently studying an oral agent that we think is going to maybe be a positive trophic factor inducer. There are so many new molecules in development that are exciting, especially based on the protein hypothesis. Remember, nothing we've done so far has touched on this protein hypothesis, which I now believe is the leading likelihood cause, that and the mitochondrial issue. So all this is exciting. And the other thing we've been working on is trial design that will allow us to be able to define an agent that is disease modifying. One of the real problems we have, and it's interesting, I work with such brilliant scientists, but they have no conception. They think they'll discover a molecule and then it will work or not work. And they don't understand that in the current world order, it doesn't matter what you have, we don't have a method to be able to say it's disease modifying or neuroprotective. And without an ability to get an approval, it's going to be really hard to get funding. So one of the things that's critically important, a major obstacle in this field, is developing a trial design or a package of designs, a biomarker, something that will allow you to be able to delineate disease modifying from other effects. And I think we're getting better. I think we have designs now. We're working with the agencies. We're using adaptive designs, which allow for much more flexibility. And I think we're in a partnership with the agencies because they want this too. But it has to be done to a certain rigor, and otherwise it's not going to happen.
Right. So in terms of young people starting in the field, you've had a lot of experiences and in this whole area of movement disorders and whatnot. What's the sort of advice that you would give them now starting out?
Go into the field. This is a great field. And I would really encourage you to go in with your heart and go in with passion and not be distracted by all of the peripheral things, especially in an era where I have absolutely no idea what the environment for healthcare is going to look like in 10, 20, 30 years. So if what you're doing is looking for money, if what you're doing is looking for power, I can't guarantee you those will be there. But if you do this with love and with passion, that I can guarantee you will be there. So you may end up poor, but at least you'll have been happy that you did something you enjoyed. And it sounds trite, but I just think it's so critically important. Here we are, you and I, years and years and years down the road, and we're as excited today as we were when we first started. And I think that's the crucial message.
Yeah.
And one of the things I tell young people in my group, in movement disorders, is you can be a clinician and you can see and observe brilliant phenomena. You can translate that or pass it on to others who will study the clinical aspects of it, who in turn can pass it on to translational researchers who can try and understand why this is happening, who can work with basic scientists to understand better the phenomena and how to treat it, who will work with experimental interventionalists who will try and find a way to translate that into more effective therapies, trialists who will do the studies, and back to the clinician who will use them and implement them. That's the cycle of movement disorders as I see it in the discovery mode. And I don't care where you are on that circle, just be on that circle.
And the energy that you describe for that is love and passion. Right?
Don't you think?
I agree.
And funding.
Warren, thank you very much. It's been a pleasure talking to you. I
You
think this has been very useful for understanding about your own career and for younger people as well.
Thank you.
Thank you.